Okay, so for my mini immune series, this is lecture 3. In lecture 3, I'm going to be talking briefly about complement. So first off, we have to ask the question, what is complement? Complement are a series of proteins that are made by your liver. And so, like any protein, you go from DNA, of course, to messenger RNA, to protein. So these are made by your body, and these are made by the liver, released into your blood, and they are released in an inactive form. So they have to be activated in order to have a function. And so right now, while you're sitting there, these proteins are circulating in your blood, so they're one of the soluble mediators of the immune response. And they are part of the innate immune response. And so they do not have a specificity. They are very general, so they don't have a specific target. They can target many different types of bacteria, and they can be activated by other components of your immune system. So they are considered innate. Now, what is important to understand about complement before we discuss the different components are its function, because this will help you to understand when we talk about the cascade of activation within complement. So the function of complement really comes down to three basic things. You're going to have a anaphytoxin function, and an anaphytoxin is basically a recruitment. So this is recruitment of immune system, and it increases is pro-inflammation. And so that's the first function of complement. The second function of complement is that it's going to help your macrophages eat things. So for example, if you have a bacteria infection, it'll bind to the bacteria, and their macrophage will be better capable of eating that bacteria. So that is called opsonization. And then the third function is the membrane attack complex. And the membrane attack complex is just what it is. It will bind to a membrane, preferably a bacteria rather than your cell membranes. It'll bind to a bacteria, for example, and punch a hole into the bacteria. And that, of course, will lead to an influx of water, and eventually membrane integrity will be compromised. And so these are the three functions of complements: amphitoxin, opsonization, and the membrane attack complex, which is also known as MAC. Okay, so now that you know the function, it's important to understand the players. So complement is a series of proteins, and they've been given the names, which are numbers. So they all start with a C for complement, and so you have C1, C2, C4, C3, C5, C6, C7, C8, and C9. And they're not written in order because C4 and C3, with respect to the cascade, are out of order. They were numbered basically how they were discovered. And so C1 was discovered first, of course, and C9 was discovered last. And so just to put the name to the function, these complements are going to be activated, and when they get activated, they're enzymatically cleaved. And so the ones that get cleaved are C2 will become C2A and C2B. C4, C4A, C4B. And the same thing with C3, C3A, C3B, and C5, C5A, and C5B. Now, you'll see that I listed the A's together. These three are the anaphytoxins. Okay, and so those are the pro-inflammatory recruitment complement components. The B's here, the two B's that have effector functions are C3B. C3B is an opsonin. And C5B forms with C7 through, or C6 through C9, the membrane attack complex. So that's the MAC. And so these are the effector functions of complement. Now, the other components, the B's, they do have enzymatic functions, which I'll discuss on this next page. So this is a diagram that I made to show complement classic pathway. And the reason I'm showing the classic pathway is because the classic pathway is the only one that uses all the complement components. So here we have C1Q, C1R, and C1S. Now C1Q, R, and S is where the classic pathway starts. So they have to be activated first. And then once C1Q, R, and S are activated, and they're activated by the acquired immune response by an antibody. Once the C1Q is activated, it will cleave C4 into C4A and C4B. C4B does not have an effector function, but it does have an enzymatic function. Complement factor 1 will also cleave C2A into C2B and C2 into C2A and C2B. C2A and C4B form an enzyme that can then convert C3 or cleave C3 into C3A and C3B. Once C3 is converted, you get that opsonization by the C3B, and then C3B can also form what's called a C5 convertase, which is basically an enzyme that can now cleave C5. And so it'll cleave C5 into C5A and C5B. C5b, of course, will go on to form the membrane attack complex with C6 through C9. And this is the MAC complex. Your anaphytoxins are going to be the C4A, C3A, and C5A. Those are all going to be the anaphytoxins pro-inflammatory recruitment of inflammatory cells. Okay, so this is how it happens. You get activation of your C1s. They then can cleave C4 and C2. C4 and C2 can combine to cleave C3. C3 combines with 4 and 2 to cleave C5. And then now you have all your effector components. You have your C3b, which is an opsonin. You have your C4a, C3a, and C5a, which are pro-inflammatory and recruitment, so anaphytoxins. And then you have your membrane attack complex, or your MAC. Okay, so just a quick word about activation of complement. Activation of complement can happen in 3 ways. So the first way we just talked about is the classic, which it requires antibodies. And there are only 2 antibodies that can do this, IgM or IgG. Okay, and then the second pathway is referred to as the lectin pathway. And the lectin pathway is based on sugars. So it's based on mannan binding proteins, MBPs, mannan binding proteins. And these are basically binding to certain sugars on certain bacteria and will activate the lectin pathway. The lectin pathway uses C2 to C9. Whereas the classic pathway, of course, we just saw uses C1 to C9, so it uses all the components. The lectin, the mannan-binding proteins are only going to start at C2 and C4. And then the third pathway is the alternative pathway. And this is going to start with C3b. And so it's important to note that C3 can spontaneously become C3A and C3b, and this can be spontaneous. And the reason for this is in an aqueous environment, these can get cleaved. Now, it's important to have some C3b around just in case you do have a bacterial infection, because then the C3b can start the opsonization process immediately. However, you don't want C3b landing on your own cells and causing a macrophage to come along and eat a nice, healthy, normal cell. So there are checks and balances within the complement system. And so I just want to speak briefly about the regulation of complement, because the regulation of complement really occurs, again, on 3 different levels. Regulation can occur at the level of C1, so this is within the classic pathway. And so you have C1 inhibitors, and that's going to C1 inhibitors. And this is, of course, going to inhibit C4, C2 activation. And so everything downstream of this, which includes the the C5a, C3a, etc., will be inhibited. The next one you want to do is you have a soluble membrane-bound inhibitors of C3. And so this is to protect against that spontaneous C3b formation, and you don't want the C3b to bind to your own cells and become activated. And then finally, there's also going to be prevention of your MAC complex. And for the same reason, just if there's no need, you don't want your membrane attack complex attacking normal cells. So these inhibitors, you have to think of them as being in balance. So your liver is going to release complement components as well as the regulators of complement. And so they're going to be at a balance. So as long as you have a normal level of complement, your inhibitors will be able to prevent spontaneous activation of C3, for example, or any spontaneous activation of your MAC complex. Now, when you have an infection, of course, during an infection, your complement components, which is the C prime, increases, where your regulators do not. So you're going to have just a constant stream of regulators, but you're going to have more of your complement components. So just the activation by your a bacteria, for example, of complement will overwhelm the regulators, but you want it to because the bacteria are there, so you want your complement to actually be working to try to control that bacterial infection. And then after the infection goes away, your levels of complement go back down to normal, and there you go into balance again with your regulators. Okay, so that's our brief introduction to complement, and we'll talk more about that in class.